In the mouth, the carbohydrate begins its chemical digestion. The salivary glands release the enzyme salivary amylase. This enzyme begins to break down the polysaccharides in a carbohydrate. The saliva itself helps to chew and moisten the food. The carbohydrate then passes through the esophagus and into the stomach. Once in the stomach, the mixture from the salivary amylase is known as chyme. The digestion of chyme is stopped due to the stomach acid stopping the effects of salivary amylase. After passing through the stomach, the chyme enters the small intestine. The pancreas releases an enzyme known as pancreatic amylase. The pancreatic amylase breaks down the polysaccharides into disaccharides, a chain of two sugars linked together. The disaccharides are then broken down by the maltase, sucrase, and lactase enzymes produced by the small intestine. This process turns the disaccharides into monosaccharides. The monosaccharides are single sugar molecules which get absorbed into the body through the walls of the small intestine. Anything from the carbohydrate that does not get digested by the small intestine, such as fiber, goes into the large intestine to be partially broken down and excreted. A lipid is defined as a fat-like molecule that isn't able to be dissolved in water. For that reason, the digestion of lipids is more difficult than other molecules. Once the food particles are chewed and swallowed, the lipids begin its journey of digestion. Fat travels in larger clumps morphed together. The lipid travels all the way down the esophagus, continuing through the stomach, and even reaches the small intestine before beginning the digestion process. While in the small intestine, bile, sitting in the gallbladder, mixes with the lipids. The bile salts will then emulsify the lipids, meaning break the small pieces of fat into smaller droplets. When the lipids are mixed and broken down by the bile salts, they will then become fatty acids and monoglycerides. The products of the lipids, such as fatty acids and monoglycerides, are then absorbed into the walls of the small intestine. The remainder of which that is not absorbed will be secreted through the bottom half of the large intestine. Nucleic acid, also known in forms of DNA or RNA, is absorbed from the chromosomes of living things we've eaten. While eating the living organisms, we have ingested the nuclei of the cells. The goal of digesting nucleic acid is to be able to digest the DNA and RNA to use those to our advantage in our bodies. The nucleic acid digested through the mouth travels down the esophagus and moves through the stomach until reaching the pancreas. The pancreas then releases two enzymes, ribonuclease and deoxyribonuclease, which then begins the breakdown of DNA and RNA. The brush border enzymes along the edge of the small intestine also aids in breaking down the nucleic acid while the molecules travel through the nucleic acid and then morphed into different monomers such as pentose, phosphates, and nitrogen bases. These components are then absorbed into the digestive capillary beds of the lining of the intestine. And again, whichever is not used throughout that absorption process is secreted through the large intestine. Proteins are made easier to swallow by the salivary glands in the mouth. The proteins pass through the esophagus and into the stomach. The stomach's gastric juices such as enzyme pepsin and hydrochloric acid begin the real breakdown of protein when the hormone gastrin triggers their release. Pepsin changes the protein into smaller and smaller pieces. The stomach's contractions form a mixture out of small pieces known as chyme, which then makes its way to the small intestine. The small intestine releases many enzymes and is where the majority of the protein digestion occurs. The pancreas goes first and then releases the enzymes chymotrypsin and trypsin in the form of digestive juices. This further breaks down the protein into peptides. The small intestine then lines its walls with carboxypeptidase, which then splits the proteins into separate amino acids with the help of the enzymes aminopeptase and dipeptase. The contractions of the small intestine help to mix and push through this process. ATP is used to transport the amino acids into the bloodstream where it is used throughout the body. A portion of the amino acid glands in the liver where it is distributed further for the last time. The nitrogen in the amino acids create ammonia, which is toxic to the body. The liver turns the ammonia into urea, which is transported to the kidney and excreted as urine.